And we're live.
All right, welcome back, everybody. So our second speaker of the afternoon session today is Dr. Ian Charest. So Ian is a cognitive neuroscientist. He's the director of the Charest Lab at the University of Birmingham, and the Center for Human Brain Health. He held a postdoctoral position at the University of Cambridge, where he studied object vision and population code representation. His research investigates the neurobiological basis of individual differences in personality and behavior, and with particular interest in human perception, memory, and decision-making. He uses a combination of neuroimaging, electrophysiological, and behavioral methods to understand the neural dynamics of representational idiosyncrasies. So today, Ian is going to talk to us about semantic representations of natural scenes in the human brain. And without further ado, the floor is yours, Ian.
All right. Well, thank you to Maine and the organizing committee for having me present some of the work that we've been doing in the recent past in my lab. So the title of my presentation is Semantic Representations of Natural Scenes in the Human Brain. This is work that I've done in collaboration with many people, including Kendrick Kay at the University of Minnesota. I'll tell you a little bit more about that in a minute. But I'm just flagging here that from May 2021, I will actually be moving to Montreal, starting a new role at the Université de Montréal, at the Cerebrum as assistant professor. So I'm very much looking forward to joining the scene in Montreal. Okay, so as a vision neuroscientist, as a vision scientist, when I see a natural scene like this one, I could think of many words or categories that are depicted in this scene. There's a vehicle, there's clouds, a blue sky, and so on. But I could also come up with a sentence description that captures the essential of this natural scene, that captures the richness of this natural scene. And so something like a yellow bus driving on a country road on a beautiful summer day. And this brings me to talk a little bit about semantics. So first, what is semantics? Well, the official definition is it's the branch of linguistics that deals with the study of meaning. And here, in the context of this presentation, I'm going to be talking more about how does the brain transform the image of the bus that we've just seen from its retinal inputs all the way up to this meaning, to this conceptual meaning, allowing me to describe it with a full sentence and communicate with others what that image was. So in other words, how is semantic information represented in the human brain? What are the mechanisms that guide this process? This is a difficult question to address. There's some previous work that have led the way in trying to understand brain representations of semantics, but you see some conflicting evidence in some cases. A lot of the work has been done with clinical populations and case studies, and it's sometimes difficult to disentangle whether the lesion could have played a role in an absence of a finding, and so on and so forth. But we're really fortunate here because I had an early access to the natural scenes dataset, a dataset that will soon become openly available to everyone. This was a tremendous effort led by Kendrick Kay and Thomas Naselaris with Emily Allen and Yihan Wu. But what is the natural data set? So it's a very, very large 7 Tesla fMRI data set where participants were asked to do a continuous recognition task. The data preparation was very well prepared. The data are incredibly rich and have a very good signal-to-noise ratio. And as I said, you can check out the naturalscenesdataset.org website to see where we're at with releasing the data to the public. But in a nutshell, the natural scenes dataset is 8 participants that have seen 10,000 images each, with 3 repetitions per image. 9,000 of the 10,000 images for a given participant were particular to him, so presented only to him, and there's a common set of 1,000 images. So that makes up roughly 73,000 images of natural scenes presented in 7 Tesla fMRI and measuring brain activity patterns at a very high resolution. So this is a scale that is completely unprecedented. So what are we going to do? We're going to be using a representational similarity analysis of semantic embeddings. So I'm very glad to follow Alona's talk, because some of the concepts that she introduced very well make sense in this talk as well. So the stimuli shown in the natural scene data set came from the Microsoft COCO database. And in the COCO database, there's 5 human observers that have written sentence descriptions of every single image. So we have these sentence caption for the experimental stimuli that came with the natural scenes data set. So to give you an example, here we have a given stimulus, and the sentence caption by one of the observers was a young boy sitting on a bed with a lamp on beside it. So using machine learning and natural language processing, we can transform these sentence captions into sentence embeddings. There's different flavors of sentence embeddings, and we've tried a few of them. And the references are at the bottom of the slides. Now, this sentence embeddings could be a 500-dimensional vector, for example, which would place the sentence, the embedding, in a multidimensional space with 500 dimensions. And for every given stimulus, we can place this stimulus, the sentence description, in this embedding space. The key here is to compute the distance between the 2 embeddings for these 2 stimuli. Once we compute this distance, we can create a semantic representational dissimilarity matrix where all of the pairwise distances between all of the 10,000 stimuli sentence embeddings are captured. So to give you a little bit of an impression of the richness of this data set, here I'm using TSNE to project this highly dimensional representational dissimilarity matrix made from the sentence embeddings of the images that were shown to the first subject in the NSD study. So TSNE essentially projects this high-dimensional representation onto a 2D plane that best approximates the distances in the high-dimensional space. So here I'm highlighting a cluster in that 2D space, and this is to show, what is typically the number of conditions that are presented to participants in a condition-rich fMRI experiment. So this shows the richness of the natural scenes data set very well. And it also shows that the semantic representations are well behaved. Here we have some board sports, ski, snowboard, and so on and so forth. Here's another cluster with tennis players, and another cluster here with fruits and vegetables and market scenes and so on and so forth. So very good data sets, and our sentence embeddings seem to capture the semantic representations really well. So what do we do with those? we are next going to perform a Searchlight, an fMRI-based Searchlight mapping between the brain activity patterns from the subjects, the NSD subjects, and correlate the RDMs in every single Searchlight location with the semantic representational dissimilarity matrices that were constructed from the sentence captions. So here I'm showing you the first subject on a cortical flat map. And so here is the back of the brain. It's cut along the calcarine sulcus, so you would have V1 here, V2, V3. I'll zoom in a minute. Don't worry too much about the anatomy at this moment. Here are all the other 7 subjects. And what you can appreciate already is how the maps that come out from the Searchlight procedure are highly similar across our 8 subjects. And we've done some statistics, and the effect sizes of these correlations are very, very strong. Given the amount of data that we have, this is not surprising. Here is a zoom in on the group average, again on a cortical flat map. So we've observed using our Searchlight mapping procedure a core semantic network, including regions of the inferior frontal cortex, the temporoparietal junction, the medial temporal lobe, the visual ventral stream, and the angular gyrus. And those are regions that often come in previous manuscripts talking about semantic representations in the brain, but for the first time, we kind of have like the whole core semantic network here in this study. All right, so next we want to better understand what kind of mechanisms could lead to this core semantic network. How does the brain, transform, as I said, the input into this rich semantic description? So we're going to turn to artificial neural networks, and this is work in collaboration with Tim Kietzman and his team, Hannes Merer, Courtney Sporer, and also Nico Kikeskotte contributed to some of this work. First, we're going to use a data set of images that has 565 categories that were selected based on frequency of English nouns and concreteness ratings of English words to come up with 1.5 million images in total from these 565 categories. And this data set is good because it captures very well the sort of visual diet that we're exposed to living our lives. So we're going to train a recurrent convolutional neural network using this image data set, using EcoSet. And this is a network that also includes lateral connections, so some form of recurrence. And we'll see, how important that is in a minute. So we're going to do the same RSA trick. We're going to send our stimulus through this RCNN, and for a given layer and a given time point, we'll have some unit activations, and we can compare for different pairs of images the unit activations, and it gives us a distance between the representational patterns for a given image in a given layer in a given time point. And we're going to compare these RCNN RDMs to our fMRI Searchlight brain RDMs. Now, here on the right, I'm showing you the result from the last time point of the last layer before the output layer of this RCNN for the 8 subjects in the NSD study. And again, you can see how consistent those maps are across the 8 subjects, but also you'll notice how similar it is compared to the semantic model fits. As a matter of fact, here I'm toggling the semantic group average again. And if I go back and forth between this semantic group average and the RCNN group average, you see how very, very similar the relationship between these models to the brain data is. Now, one cool thing that we can do with the RCNN, given that it has these recurrence and lateral connections, we can look at sort of representational dynamics and track the representations across time points across the layers of the model. And we see this nice progression from the back of the brain to high-level visual ventral areas, including the fusiform gyrus and the inferior temporal cortex. And finally, in this, what I call the core semantic network of brain regions, very similar to what we saw with our semantic model. Now, with Tim, Tim developed another model, which is called VNET, which he's preparing at the moment. And this model is a model that's kind of more inspired by the brain. And we've added in this project kind of additional constraint. Instead of training it on category objectives, we trained it on semantic embeddings. So VNET, essentially, Tim and his team have done incredible work to try and match the receptive field sizes across the layers of the model to known biological receptive field sizes in the literature along the visual ventral stream. And we used the same trick again. Searchlight-based fMRI RDMs were compared to layer-based RDMs in this model for our stimuli. And here I'm showing you the correlation map. And again, we see an even better fit to our core semantic network with correlation values approaching what we saw when we used the semantic RDMs that were created from the sentence descriptions. So far, we've seen that we can model the brain patterns and sort of see these semantic brain representations, which suggests that there's sufficient information about the semantics to try and reconstruct scene descriptions from brain activity patterns. And this is what we tried next. So it's fairly simple. We have sentence embeddings, as I said, for the stimuli presented to our participants. And we're going to try and find a set of linear weights that applied to the fMRI patterns combined with an error term enables us to predict the sentence embeddings. Now, once we've found those linear weights, we can apply them to left out fMRI patterns from images that haven't been used in training to find the linear weights. And this will give us predicted sentence embeddings. And now, once we have the predicted sentence embeddings, we just use a lookup strategy. So we have 3.1 million Google conceptual captions and their embeddings. And we're going to try and find from the predicted embeddings which of the 3.1 million captions best resembles the predicted embedding for a given image. And this is going to give us with decoded sentences for the left out images. So here I'm showing you a few examples. The predicted sentence for this image is boy hitting the ball while playing tennis. And so this was predicted from brain activity data. Here is a man with the surfboard checks out the surf. A girl child is learning to ski. A young man jumping high on a skateboard in a skate park. Tree-lined roads and railways are common on the map. So some of the predictions are not quite there, but this is not that far from what we're seeing. A herd of cows grazing in a field with cow very pregnant and due to calve any time. This is too accurate. And finally, pizza. Let's dig into this inch dish of slices. So Next, I want to talk a little bit about some of the work that my student Daniel Lind has been doing on a slightly different topic, but I think it will follow well from what we've seen so far. So Daniel has been working on visual consciousness using paradigms like the attentional blink to modulate conscious perception in our participants. And his framework is essentially using RSA to characterize the representational geometries either in models or in brain or in behavior, and try and predict conscious access to the stimuli that were presented to the participants in the attentional blink. Some of his work is published already. I'm not going to talk about the deep learning side of it because that's published in Nature Communications in 2019. This is a follow-up study. I'm going to present you a couple key results from it where we used brain imaging, fMRI, and EEG to try and predict conscious access. So first, when we look at the inter-target distance that we measure from the brain activity pattern, so this is using the fMRI data, we see a network of brain regions that is highly similar to the core semantic network that we've seen so far, that predicts the likelihood that participants will detect the second target in an attentional blink trial, depending on the distance of that target to the first target in that attentional blink trial. And second, and this is really cool, Daniel can explain the sort of a little bit remarkable phenomenon of the individual differences in attentional blink. Some participants are called non-blinkers. And Daniel has done this Searchlight analysis where he looks at representational richness in a given sphere of the Searchlight simply by averaging the activity patterns, the distances between the activity patterns for all pairs of images, and then correlates this with the overall performance in the attentional blink task. And he sees this right lateralized network, including the temporal parietal junction and regions of the visual ventral stream and inferior frontal regions that predict really well a participant's performance in the attentional blink. So possibly some elements of answer to the individual differences in visual consciousness. And the final thing I want to talk about is work that has been done by another student in my lab, Simon Fagel, who's been using electroencephalography to look at semantic representations in the brain. One of the plans I have when I moved to Montreal is to try and extend the natural scenes data set with electroencephalography data. And Simon did some first steps in this direction, looking at semantic representations in the brain. But this is a particular data set because Simon recruited participants that are referred to as super recognizers. So these are participants that have incredible ability to recognize faces. And he's also recruited a group of matched controls. And what is the first thing that we noticed, simply giving the topographies from the EEG to a linear classifier, trying to predict group membership, so whether a given participant is a super recognizer or not, we can do that with great accuracy. And this doesn't matter whether we're using trials that showed faces in natural scenes or non-face trials. And next, we can do with EEG representational similarity analysis the same way we do it with fMRI, but this time we do it in time using the EEG channels as features to estimate the representational geometries. So we'll have RDMs for every single time point. And we can correlate these RDMs to semantic representational dissimilarity matrix, which again was computed using sentence embeddings of sentence descriptions of the stimuli that were shown to the participants. And what I'm showing you here is on the y-axis, the similarity between the semantic RDMs and the EEG RDMs. And here, notice Simon did a control for low-level kind of properties using the first convolutional layer of deep network. So this is a partial correlation. And what we noticed at first glance is better correlation in the super recognizers early on, before 200 milliseconds, and also this kind of bump around the P300s, lasting all the way until 700 milliseconds. Earlier peak latency for the super recognizers compared to the controls as well. So this is interesting because it suggests that the incredible perceptual ability of the super recognizers is related to semantic representations. So I'm going to stop here and conclude. So I hope to have convinced you that the natural scenes dataset is extremely valuable to the community, and we can, with it, do very precise and rich representational modeling. Using sentence embeddings, we were able to create semantic models and reveal a core semantic network in the human brain. Using our artificial neural networks, we were further able to see the sort of representational trajectory in the brain from early visual areas to late visual areas and the core semantic network. Training VNET on semantics led to very, very good performance in predicting semantic representations in the brain. We have a few links to behavior here, one with visual consciousness and another with perceptual ability in our super recognizers, where the dynamics of semantic processing seem to be contributed to these differences in ability. But overall, the results that I've shown you today provide a broad view of how the visual system transforms sensory input into high-level representations most relevant for cognition and behavior. And I would say it tries to go one step further from the sort of classical categorical account of high-level vision and bringing it to a more linguistic, conscious space. One more thing, most of the behavioral experiments that we have done with this in this presentation come from our website Meadows, and my postdoc Jasper van den Bosch is working really hard to make Meadows a really fun and seamless experience for experimenters to conduct behavioral experiments online. So perhaps valuable in this time of COVID-19. And I would like to thank the collaborators on the various projects in my lab and the funding bodies. And I would like to thank you for your attention.
All right. Thank you very much, Jan. This was an amazing talk. Very impressive results in there. We have some time to take a few questions. So there's some coming in online here. So let me read you some of them. So the first one from Federico Micheli asks, interesting results about consciousness. How did you define representational richness? So it's kind of a
yes. So yeah, I mean, I think there's several ways one could go about trying to define representational richness. Here, we just looked at a kind of metric that would index some kind of pattern discriminability. So how well can we separate brain activity patterns in response to the different exemplars shown to the participants? So the very simple solution was to average the upper triangular part of the representational dissimilarity matrix, averaging all of the pairwise pattern distances into one score, which is kind of the representational richness score. And this is what we then correlate with the sort of performance of a participant in the attentional blink task to reveal this, this network of brain regions explaining the individual differences.
Very interesting. Okay, but you could argue that there's other ways that you could go about to measure this to a certain extent, right?
Yeah, absolutely.
Okay. Very cool. So moving on to a question by our very own Panda, aka Karim Gerbi. He asks, you use the raw amplitude of EEG calculations, i.e., for the individual time bins. Did you try using band limited amplitude, example alpha or beta amplitude?
Yeah, this is a very, very good question. I've started playing with band limited amplitude, so essentially decomposing the signals in the different frequency bands and so on, prior to training classifiers. So I've tried this on other data sets before, but we have not yet applied it to this data set with the super recognizers. So for this particular data set, I don't have a specific question. It would be really interesting to see if our semantic RDMs, if we could constrain the sort of oscillation profile that best capture them. But my experience with band limiting is that you're often not going to get as clean decoding as when you just give it the full spectrum of the data, basically.
Okay, very cool. There's one question here from Brad Wibble, or Weibel, I apologize if I pronounce your name incorrectly, which I shared actually. The question, how were non-blinkers identified, right? Using the same behavioral data as in the experiment or with an independent data set?
Yeah, so this is a good question by my friend Brad. The attentional blink task was done while we measured the EEG, and this was done outside of the fMRI scanner, so this wasn't a sync study. So participants were doing the attentional blink task in EEG with the same stimuli that we later present to them in the fMRI. And the task in the fMRI is not an attentional blink task because we're not crazy. I think it would just go too fast. Maybe it's possible. I kind of dream about doing this one day. So the task is a very simple working memory task, which we think can sort of engage the steps of processing that ultimately leads to visual consciousness. So the data are independent in that way. It's from the same participants, but from two completely different experimental sessions, one while we measure EEG and another whilst the participants do this working memory task in fMRI.
Very cool. Very cool. All right, there's another question here, quite interesting, from Cleona O'Doherty. Is the ANN models, were these entirely visual, or did they have linguistic components? To what extent do you think that meaning demands language, or can it be extracted from purely visual input?
Well, I think it's all linked. The fact is we very rarely have a solely visual experience. We communicate a lot. We communicate about what we see. But specifically for your question, the recurrent convolutional neural network was trained on a kind of classic category objective. But the VNET that I showed later on was trained on fast text embeddings that were computed from the sort of category words of the objects that are depicted in the images of EchoSet. So it was a different objective function where instead of just trying to predict the category of a scene, we're trying to predict a representational embedding that comes from a linguistic representation.
Very cool. All right. So I think we have one last question here, although there is, actually, before I go to that last question, there's a follow-up by Brad. Have you tried to link these visual semantics representation to auditory activations?
Not yet, although we see some activity in high-level auditory cortex, some links with semantics in our visual task, which I find fascinating. It would be really, really, really fun to do an auditory experiment and present the sentence, spoken out to the participant, and maybe also having the same participant trials where you show the scenes where these sentences came from, and then start comparing all of this data. Absolutely.
Very cool. And last question, very rich and high dimensional data set. When can we expect the release? This is a question from Rishab.
Yes, I think I'm not exactly sure of the official date. I know Kendrick is working really hard to try and get the data prepared and ready. I would say probably in the new year sometime. I don't know if it'll be January or a little after that, but it's worth waiting for. It's an amazing data set.
All right. Cool. So we will stay tuned then. Well, thank you again, Jan. And we will now take a little changeover break. We'll be back in just a few moments.